Now you should be able to see the worksheet. Do you?
Yes.
Worksheet Mendelian genetics.
Yes, sir.
Okay, so let me just go over the answers, at least the answers I was looking for. Gregor Mendel was the first to demonstrate the existence of genes, despite many experiments in breeding that had preceded him. What did he do that no one else had done? Two things. He first had inbreeding exercises where he developed true breeding, completely homozygous lines. That's one thing. The other thing is, instead of stopping with the F1 generation, after he did his first cross, he went to the F2. He took two individuals from F1, crossed them with each other, and got an F2. And it's only in that way that he discovered recessive characteristics. They only show up in the F2. So that's a very important point, two very important points of what enabled Mendel's success. The next one, Darwin didn't know Mendel or his results, and yet he established the theory of evolution based on natural selection. What's the relationship between the contributions of these two people? Well, Mendel's laws of inheritance provide the rules, provide an explanation for Darwin's concept of natural selection. And that's what allows evolution to proceed. So Mendel's laws rationalize what Darwin couldn't explain. He hypothesized that natural selection was a force, but he didn't have any basis, any molecular basis for saying that. Mendel's laws provided it. Distinguish phenotype from genotype. Phenotype is the outward expression of traits, what you see with your eyes. Genotype is the genetic makeup, and it's expressed as symbols, usually letters. How might inbreeding depression lead to hybrid vigor? So we talked about inbreeding depression. Very often when you start a series of inbreeding crosses, the progeny of each succeeding cross keep getting weaker and less vigorous. But in the end, if you cross two inbreds and look among the progeny of various crosses, you will usually find that certain crosses between certain inbreds will show hybrid vigor, which means more robustness than either of the starting parents. And I showed you the, excuse me?
What is progeny?
Progeny are the children. You remember I showed you the example of hybrid corn, where the hybrid was this huge tall plant compared to the parental lines and produced an ear about twice the size of the parental. So hybrid corn is a classical example of hybrid vigor and how the only corn that's grown these days is hybrid corn. And corn seed companies keep, they never patent a genotype or an inbred. They're trade secrets. So they never allow to escape from their company the names of the inbreds or the genotypes of the inbreds that are good, that give them their best hybrids. Okay, self-pollinating plants are naturally inbreeding. Why is that? Well, it's because the plant fertilizes itself. So when you're trying to do a self or an inbreeding generation, you cause the plant to fertilize itself. Self-pollinating plants do that naturally. The plant's pollen is the male gamete, falls on its own stigma, which contains the female gametes. What is the technique for making a cross between two self-pollinating plants? Well, we looked at what Mendel did with his peas. Peas are self-pollinating. In fact, before the flower ever opens, pollen sheds on the stigma. So what Mendel did was, before the flowers open, he dissected them manually and took the anthers out, so there was no pollen. And then later, he would come back to the same flower with pollen from a different plant and put it on the stigma. So as pea flowers are not very big, and this was a lot of very tedious work because he did thousands and thousands of crosses. How would you distinguish a dominant trait from a recessive one? Well, you cross inbred lines representing the two traits and see what appears in the F1. The F1 should be all alike. Whatever that phenotype is, that's the dominant trait.
Oh, did you come in the garage?
What?
Sorry, nothing.
Okay. All right. I thought I heard a parrot. Okay. You have a set of plants described as having either purple flowers or white flowers. And another set is described as having flowers that are either big P, big P, big P, little p, or little p, little p. Which of those designations is the genotype and which is the phenotype? That should be obvious to you. The genotype is the letter symbols, the phenotype is the words. Now this next one is sort of involved. It has a long answer. A tall pea plant is inbred for 6 generations. A short pea plant is also inbred for 6 generations. And then a 6th generation tall plant is crossed with a 6th generation short plant. What will be the genotypes and phenotypes of the F1 and the F2s? Based on your result, which trait is dominant? So you have to think about this, and this is could be representative of a question that might come up on a test. So to answer this question, you first have to determine the phenotype of the F1. So you do an F1, and if in the F1 all the plants are tall, then tall is the dominant trait. And you can give it the gene symbol big T. The alternate trait would be short and is therefore recessive, and would be given the gene symbol little t. So the genotype of the F1 would be big T, little t, and the phenotype would be tall. In the F2, the phenotype would be 75% tall plants and 25% short. And the genotype would be 1 to 2 to 1, 25% big T, 50% big T, little t, and 25% little t. So that's one possible outcome. However, if the alternative happened, if in the F1 all the plants were short, then short is the dominant trait. And it can arbitrarily be given the symbol S. Arbitrarily means you can choose any symbol that you like. I chose S, big S. The alternate trait in this case then would be tall and is therefore recessive and would be given the gene symbol little s. So the genotype of the F1 would be big S, little s, and the phenotype would be short. In the F2 generation, the phenotype would be 75% short, 25% tall. The genotype would again be 1 to 2 to 1. Big S, 50% big S, little s, and 25% little s, little s.
Question, please.
So is it clear how you think through a question like this?
No, could you explain it just one more time? I'm still like kind of confused.
Okay.
I have a question too, sir. What's the sample for genotype?
Say that louder.
Okay, what's the sample for genotype? Because I have now you say the phenotype is the sample is S, right? A capital S. And genotype it should be little s.
No, the genotype is S. If you're talking about the dominant allele, it's big S. If you're talking about the recessive allele, it would be little s. That's in the case that when you did your cross, all the plants in the F1 were short.
All right.
Okay, and then someone asked to go through it again. Okay, so you have these two inbred lines, inbred for 6 generations, each of them. So you know that they're homozygous. And you cross them. One has a phenotype tall, the other has a phenotype short. And so one possible outcome when you cross them is that in the F1, all the plants are tall, like the tall parent. In that case, tall is the dominant trait, and you can give it, and again, arbitrarily, I chose the symbol T. Big T is the dominant trait for tall. And so the alternate would be short, which would be recessive. It doesn't show up in the F1. And it would be given the gene symbol little t. Big T for dominant, little t for recessive. And so the genotype of the F1 would be big T, little t. So is that clear? Why the genotype would be big T, little t?
Yes, sir.
Yeah, thank you.
And the phenotype would be tall. And in the F2, it's when you get segregation. So the phenotype would be 75% tall, 25% short. And the genotype would be 25% big T, 50% big T, little t, and 25% little t, little t. And it's only the little t that would show the recessive trait, which is short in this case. The rest of the answer is just, what if the opposite happened? What if you did this cross between the two inbreds and all the progeny in the F1? Progeny is children.
Professor?
Yeah?
How did you get those percentages?
I wish I had last week's. Let me see if I can get last week's slides up here. You know, it would be easiest to explain it that way. Okay, I suspect you can't see a new screen. Is that right? I'm going to have to stop sharing and then reshare. Can you see the screen that says monohybrid cross?
Yes.
Okay, so that's what this is. In this case, instead of using tall and short, we're using flower color. Purple and white, and purple is dominant. So the question was, how do you get those ratios? If you go to the bottom here, you see that if you start out with a dominant big P parent and a recessive little p parent, all the gametes produced by big P are big P, and little p, they're all little p. And so when you cross them, every plant is heterozygous, big P, little p. So the F1, they're all alike, they're all purple because they all have the dominant allele, big P. But then when gametes are made, half of them are big P and half are little p. So then you see the segregation in the F2 at the bottom. So the phenotype is 3:1. That's because anything that gets a big P allele is purple. And only if you get 2 little p alleles is it white. And so phenotype in a monohybrid cross is always 3:1 in the F2 for phenotype, and 1 to 2 to 1 for genotype. And so one big P, you see if big P egg fertilizes, or big P sperm fertilizes a big P egg, you get a big P progeny. If on the other hand, little p fertilizes big P, or vice versa, you get big P little p or big P little p. So that's 50% of the population. And only if little p fertilizes little p, you get a homozygous recessive. That's 25%. So the ratio in the F2 is 1 to 2 to 1. So does that answer your question? I mean, we're in the worksheet, I'm talking about tall and short. This is purple and white, but it's the same thing. It's the same procedure to work it out in both cases. Is that clearer now?
Yes.
Where those ratios come from?
Okay. Do you say, I mean, the percentage should be 75% for big P, right? I mean, that percent is not going to be changed or what?
The first what?
The percent for 3:1. I mean, you have the F big T and
Yeah,
big
that's
P. Yeah.
that's the phenotype, and
Yeah,
that's fixed.
yeah, sure. I mean, this percent is never change. I mean,
Yeah, in a monohybrid cross, regardless of what your characters are, assuming that your parents are inbred, they're homozygous at all loci, you will always get in the F2 a 3:1 ratio for phenotype and a 1 to 2 to 1 ratio for genotype. Doesn't matter what the traits are that you're looking at, those will be the ratios. All right, I'm going to take this away. Get back to the worksheet. Stop sharing. You should be seeing the worksheet again. Tell me if you're not.
Yes.
Yes.
Hearing nothing but yeses. Great. So we're going to leave that question and go to the next one. You have a plant with purple flowers, and you choose the symbol big P to indicate the genotype of purple flowers. And you assume that the color purple is dominant to another flower color, which is white. And you designate that as little p. So you have big P, little p, distinguishing flower color. So your purple flower plant could have the genotype big P, or little p. How would you determine which it is? Well, the answer is you'd perform a test cross. And we talked about test cross. Well, I don't want to switch screens again, unless somebody wants to see it. But I showed you a test cross last time. And so what you would do is cross a purple, which has a genotype of either big P, or big P, little p, times a white plant, which is homozygous recessive. You know it's homozygous recessive because it's white. So it has the genotype little p. And you look at the F1. If all the individuals in the F1 have purple flowers, then they all have the genotype big P, and the genotype of the unknown is homozygous big P. If, on the other hand, in the F1, half have purple flowers and half have white flowers, then the purple half have the genotype big P, and the white half have the genotype little p. And the genotype of the unknown is heterozygous, big P, little p. But it's important that you know what a test cross is and how you would do one. Is this clear? If it's not, I'll switch screens again and show you the example on a slide. Or Scott, maybe you can explain it better than I have.
The test cross?
Yeah.
Si te quieres, nos vamos, bebé. Ya.
Yeah, for the test cross, you kind of have to see it done to understand why it works, but you have to have a true breeding recessive to cross with your questionable plant to see how the traits are going to come out. And really, the best way to demonstrate it is to lay out a Punnett square, I think.
Yeah, okay.
A ver, llama, ¿qué me llevo? Porque con esto ese, because I'm not gonna
Doris, could you please mute your microphone?
Okay, I'm going to share the screen again and show you the test cross.
I think one of the key points to a test cross is you have to know that you are testing a true breeding recessive. You can't have any question about that. Because if it's not a true breeding recessive, then you uh, which means that it's essentially little p if we're talking about purple and white flowers or something, but it has to be, both genotypes have to be recessive. It cannot be a heterozygous. Otherwise, you still don't absolutely know what you're dealing with.
Yeah, and that's true. You should be able to see on your screen now a slide that says the test cross. How can we determine unknown genotypes? And you do that by taking the individual of unknown genotype and cross it with a homozygous recessive. In this case, we're talking about flower color. So you have a plant with purple flowers. You know that it has at least one big P allele, but you don't know if the other allele is big P or little p. So how do you determine which it is? You cross it to a plant whose genotype you know. And that's one with a white flower, which you know is little p, little p. So that you have two possible outcomes. If in the F1, all the plants have purple flowers, then you know that your purple plant progenitor was big P, big P. And if half the flowers are purple and half the flowers are white, then the purple plant that you began with, that you're testing, must have been heterozygous, because it's half of its gametes turned out to be little p. That's how you get white flowers. Was that clear about a test cross? Okay, if there are no more questions about it, I'm going to stop sharing that.
Professor, do you know, will we have any time today during class to maybe work some of these problems out? Just as like a practice, or is that something that you intended more for homework and workshop?
Well, that's the default. If we have time, after I go through the molecular genetics stuff, yeah, I'd be happy to have you do. You have some problems already set to go?
Yes, sir. If you think we'll have time, I will have a couple problems ready to go.
Okay. Well, I don't know if we'll have time.
I will look.
That's right.
I will look for problems, and I'll have some on standby.
Yeah. Okay. Well, one thing I can do is just make sure you have time at the end, and we can finish this lecture next time.
And also, I don't mean to take time away from the students. If the students understand this and don't feel like the practice is needed, then I apologize for speaking up. But if you guys think that that would be useful, certainly willing to find you a couple practice problems to work on in class. So let us know.
Anybody speak up if you'd like to have Scott work out some problems for you in this class period.
I would like that.
Okay. Well,
So the last 10 minutes, maybe we see if we understand or not. Maybe we can,
yeah,
use the questions. Or,
yeah. I think probably most of us could benefit from going through some of these. So I will keep track of the time. Scott, if I go past 15 minutes left, let me know and I'll stop at that point.
Aye, sir.
Okay. Going back to the worksheet. You should see the worksheet again. Okay, so we're done with the test cross, and the rest of what's on this worksheet are simply definitions, and I'm not going to go through them. You can read them all here. And also they're straight off the summary slide at the end of the slide set. You all have the slide set, so you all have the summary slide, and all the definitions are there. And you should all be able to see molecular genetics. You see a slide that says molecular genetics?
Yes.
Okay. I'm going to put it on the slideshow so you can see it better. Okay, now this is a complement to what we've been doing with Mendelian genetics, where you're dealing with abstractions. In Mendelian genetics, genes are just symbols. Molecular genetics, genes are pieces of DNA. Well, there are a number of developments in the last few years that have truly revolutionized biology and consequently much of what goes on in the world. And what I'd like to do in molecular genetics is just highlight a few of these developments, all of which were or are surprises, things you wouldn't predict. Now this, just to remind you, this is the Nobel Prize for CRISPR. We talked about that last time, so I won't take time to say anything more about that. But to talk about another Nobel Prize in physiology or medicine in 1978, it was won by these three individuals for discovering restriction endonucleases. Restriction endonucleases were totally unexpected, big surprise. And prior to, should we get rid of this? Prior to the 1970s, nucleases were thought to digest nucleic acids indiscriminately, and no one would have ever predicted that they might be site-specific, that is, cut at very precise places in the genome. None of the other enzymes that digest macromolecules are site-specific. So you have proteases are not, they just chew up proteins. Lipases that digest lipids are not site-specific. And carbohydrates that digest carbohydrates are not site-specific. But enzymes that digest nucleic acids are. Now, but they aren't all. So some are non-specific. They can be exo or endo nucleases, like other types of enzymes. And if they're on the right-hand side here, an exonuclease chews off nucleotides from the end of a DNA molecule. An endonuclease chops up in the middle, places other than the ends. A restriction endonuclease goes just to a specific site and makes a very precise cut at just one site. So restriction endonucleases, the source is always bacteria. Eukaryotes do not have restriction endonucleases. And in bacteria, the purpose of a nuclease is a defense mechanism against viruses. So when a virus comes in, the endonuclease chops it up, chops up its nucleic acid. DNA methylases protect themselves from their own enzymes by methylating the cut sites, the restriction enzyme cut sites. But there is a restriction enzyme called type 4, which in fact cuts even methylated sites. So how is a cut made? You see that here on the lower right. A cut can either be either result in a sticky end or a blunt end. On the left side of this diagram, you can see the recognition site. When it's cut, you have overhangs which are called sticky ends. So the AATT correspond to the TTAA, and that is a sticky end, and you can substitute foreign DNA with the same sticky ends into that hole in that cut site, and that's how cloning is done. On the other hand, the cut site can result in blunt ends with no overhangs. That's also useful in molecular biology, but not as much. Okay, here are the various types of site-specific endonucleases. Type 1 cuts at a specific site, but it's about 1,000 base pairs away from the recognition site. So not very useful in molecular biology. It's the type 2 restriction enzymes that are extremely useful. The recognition sites are palindromes. I'll tell you what a palindrome is in a minute. They're 4 to 8 nucleotides long. And what's important is that the recognition site and the cleavage site are the same. They're either at the center of both strands to yield a blunt end or at staggered position, leaving sticky ends. There are about 3,500 type 2 restriction enzymes known. That's a lot. It's a big tool chest. Type 3 restriction enzymes are a lot like type 1, except the cut site is closer to the recognition site. As I said earlier, type 4 enzymes recognize methylated DNA. I want to emphasize type 5 restriction enzymes because they use a guide RNA, sound familiar? A guide RNA to target a specific non-palindromic sequence. And they cut the DNAs of variable length. And a good example of that is the CRISPR-Cas9 enzyme. Once the guide RNA guides Cas9 to a site, it cuts at that site. And that's how you can target any position in the genome. And we can't leave this topic without talking about an enzyme that isn't a restriction endonuclease, and that is DNA ligase. It's a DNA joining enzyme. Remember we talked about this in Okazaki fragments, hooking Okazaki fragments together. Same thing happens in cloning. If you want to, you can put a piece of DNA with sticky ends, but it still isn't joined, and DNA ligase comes along and joins the two ends. Now, one of the sort of mysterious things about restriction endonucleases is their names. Where do these names come from? EcoR1, ND3, and so on. They come from the names of the bacteria that produce them. So for example, Escherichia coli, E. coli, produces a number of restriction enzymes, EcoR1, EcoR2, EcoR5, and others. R usually stands for the strain, and if there's anything else there, it further subdivides the source of the restriction enzyme. So Haemophilus influenzae makes HindE3, HIN. Bacillus amyloliquefaciens makes BAM, H1. Staph aureus makes SAW3A. Proteus vulgaris, PVU2. So that should demystify where names of enzymes come from. Okay, now we're going to talk about plasmids and cloning. Now, what is a plasmid? It's a circle of DNA, usually. Not always, but usually plasmids are circles. They're found primarily in bacteria, but some occasionally occur in eukaryotes. They often have functions associated with them, like encoding a protein that's toxic or an antibiotic resistance. Now, to make them useful in cloning, they need several things: an origin of replication, a selective marker, and which is usually antibiotic resistance, and a multiple cloning site, which I'll show you an example of that in a minute. I've talked about palindromes, how all of these recognition sites are palindromes. And if you go to the left, you can see in the middle here where I'm pointing, an example of a palindrome. So on one strand, starting at 5', it's GAATCC. You go to the other strand, and it's the same thing, starting at 5'. It's GAATTC. And if you look at all of these, every one of them is a palindrome. To give you a better idea of what a palindrome is, look at the bottom here: a man, a plan, a canal, Panama. Now read it backward. Do you see it? How it reads exactly the same backward as it does forward. Well, if you don't, you can think about it later. Another example: Abel was I ere I saw Elba. If you read that backward, it says Abel was I ere I saw Elba. So that's what a palindrome is. It reads the same forward and backward, and all restriction endonuclease sites are palindromes. The length of a site can vary from 4 nucleotides to 8, and that determines how many there are in a genome. So if the length is 4, there are approximately 256. A cut site occurs every 256 base pairs. If the cut site is 8, it's 65,000 base pairs between cut sites. So if an enzyme cuts at a site with 8, it cuts much less frequently. And for example, NOT1 does have an 8 base pair restriction site, and people who want to make very large fragments, libraries of very large fragments of DNA, will almost always digest the genomic DNA with NOT1. Now, I circled 2 enzymes here because for me this is personal. The first genomic library I ever made, I digested genomic DNA with SAW3A, which you can see is a 4-cutter. And I mixed it with a plasmid digested with BAMH1. Now, if you look at the restriction sites of those 2, you can see why. So I wanted very small fragments, so that's why I used a 4-cutter. And you can see that its restriction site is GATC. If you look at BAMH1, it has an overhang of GATC. So I made sticky ends on the genomic DNA. I made the same sticky ends in the cloning vector, so it went together. Okay, I said I would show you an example of a prototypical cloning plasmid. It has what's called ORI, origin of DNA replication. It has to have that in order for it to replicate in bacterial cells. It has to have a selectable marker, which is usually antibiotic resistance, and in this case, it's ampicillin resistance. And it needs what's called a polylinker, which means a short piece of DNA that has a lot of restriction enzyme sites, and they're almost always 6 base pair restriction sites. So here you see BAMH1, the one I just referred to. So these are all very commonly used restriction enzymes, and it provides the investigator a lot of options for what they want to digest the target DNA with in order to clone it into this plasmid vector. So is it clear what a cloning vector requires? So how do you actually clone? Well, you start with a plasmid, and you cut it with a restriction endonuclease, in this case, EcoR1. Then you have foreign DNA. This could be from your favorite organism. You isolate total DNA, and you cleave it, digest it with EcoR1. So now you have EcoR1 sites in your target DNA and also in the cloning vector. You mix them together, and the sticky ends find each other and anneal. So they, due to hydrogen bonding, they anneal. And then there's one final step, that's DNA ligase. These little gaps do not seal by themselves, and DNA ligase has to do that, and so you then have a chimeric plasmid that can be selected in E. coli and cloned. For the sake of time, I'm going to skip this one, and you can review it. It's pretty much a review of what I went over on the previous slide. But I want to make one other point. Okay, the main point I wanted to make here is that if you're cloning from a genomic digest, you don't take total genomic DNA, because as you remember, 95% of the genome does not code for protein, and you're looking for genes. So how do you sort out the genes from the rest of the genome that is not genes? Well, you do that by, instead of isolating DNA, you isolate messenger RNA, mRNA. But you can't clone mRNA because you can't clone RNA into a DNA vector. So what you do is use an enzyme called reverse transcriptase to copy the RNA, single-stranded RNA, to double-stranded DNA. And that's what you digest and clone in a vector and put into E. coli. Now, I've just introduced a term I don't think we've talked about before, and that is reverse transcriptase. What is it? Well, in the early days of molecular biology, there developed what came to be called the central dogma. It was inviolable that DNA encodes RNA, which translates to protein. It's a one-way street. DNA always goes to RNA, always translates to protein, until retroviruses were discovered. Now, it turns out, if you look on the right here, a retrovirus, and an example of a retrovirus is HIV, which is the virus that causes AIDS. It's a single-stranded RNA genome, and part of that genome encodes this enzyme called reverse transcriptase. So what retrovirus does is synthesizes double-stranded DNA using its own viral RNA as a template, and that DNA then integrates into the host's genome. So when the host genome transcribes its own RNA, it also transcribes virus RNA. And that viral RNA is transported into the cytoplasm where it starts making new virus particles. So that's the life cycle of a retrovirus. The important thing for our purposes is reverse transcriptase. So it's, the central dogma is wrong. It isn't a one-way street. From RNA, you can either translate to protein or be reverse transcribed to double-stranded DNA. So remember that might come up sometime as a question. All right, I think I have just about enough time to go through polymerase chain reaction, and then I'll stop, Scott, and you can do some problems.
Thank you, sir.
So what happens if you want to make a lot of DNA, starting with not very much? So polymerase chain reaction allows you to make a lot, essentially as much as you want, large quantities of a specific segment of DNA. Prior to PCR, the only way to make large amounts of DNA was grow it up in E. coli cells, harvest the cells, and extract the DNA. This can all be done in a test tube. Okay, PCR. All right, on the center and right-hand side here, it shows you how PCR is done. So you start at the top with a target sequence. So this is DNA. It could be a piece of DNA in the genome. It could be a clone that you want to make a lot of. So what do? Well, first of all, you heat your DNA sample, usually very hot, like 90 degrees centigrade, so that the strands of DNA separate. And then you cool it, and along with your target DNA, you have primers. You have to always have primers for DNA polymerase to work. You have DNA polymerase, and you have the bases to make DNA, A's and T's and G's and C's. So they're all in this mixture together. And I have to emphasize the DNA polymerase. It's called Taq polymerase, which is stable at 98 degrees centigrade. Its optimum for function is 70 degrees. It's from a bacterium known as Thermus aquaticus, which was isolated from a hot spring in Yellowstone Park. And it's absolutely essential for PCR to work, that you have to have this heat-stable DNA polymerase. So you heat up to separate the strands, and the DNA polymerase is stable to that. And you anneal, you cool it and allow the primers to anneal to their sites. And then you heat it up again so that DNA polymerase, Taq polymerase, can fill in between the primers. And so now, if you start with one molecule here, after 3 temperature shifts, you have 2 molecules here. And if you do another cycle, these 2 molecules become 4. You do another cycle of temperatures, and you now have 8 molecules. If you go over to the right, after 5 cycles, you have 32 copies. 10 cycles, 1,000. At 20 cycles, you have over 1 million. And at cycle 25, you have 33 million copies after having started with 1 just a short time before. So the procedure for that is off to the left here. You have a denaturation step, separates the strands. You have an annealing step. So the denaturation step is usually some temperature above 90 degrees. You have an annealing step, usually 50 to 60 degrees, for 20 seconds or so. And then you have an elongation step at 75 degrees, which is the temperature optimum for Taq polymerase. And that adds bases at the 3' end, as you know from going over DNA replication. And so it takes maybe 5 minutes to do the elongation step at every cycle. So what you would like to do is automate this. And so here's the equipment that you need, either microtiter plates or small reaction tubes. And it's critical to do large numbers to have a thermocycler, which cycles from one temperature to the next temperature to the third temperature and back to the first temperature again. And if you're really sophisticated, you have a robot doing it all. But again, coming back to my own personal experience, when we first started doing PCR in the 1980s, we did it this way with a water bath. So you'd stand there with your tubes and stick them in a 94-degree water bath for a few seconds, then move it to 55 for a few more seconds, then to 75 for a few minutes, and then back to 94 again. And just keep doing that for as many cycles as you want. And for example, to get 33 million copies of your target, you needed to do 25 of these cycles. So this, everybody hated having to do PCR until thermocycles came along, and that made it easy. Now, I want to tell you, okay, I'm going to stop at that point, Scott, because I have quite a long story to tell about Kary Mullis and how he invented PCR. Very unlikely event, but we'll pick it up here next lecture. So Scott, go ahead with your problems.
All right, thank you, sir. Would you be able to enable a screen share for me?
I can try. Stop share that. And okay. Share the screen with.
I think there's 3 dots in the share screen, or right click on it.
At Zoom screen, or is it whiteboard?
I'm going to need the whiteboard, yes.
Okay. Okay, there's your whiteboard and your pen.
I don't, it's you, you're the one that controls everything. I don't have.
You can't control that?
No, sir.
Okay, well, we need to have you in control, so.
Well, and either that, or if you'd like to finish your lecture, what I can do is I can just devote today at workshop to covering Mendelian genetics for folks. Going back over working problems, I will simply let everybody know that that is what workshop will be today. And anybody that can attend will be able to see, we'll work through problems for 2 hours if that's what people need. I don't mean to make things complicated on your end, sir. So if
there's any way for you to do it here, I think it would be good, because you have the entire class and you may not have the entire class for workshop.
Yeah. I can't share my screen because participant screen sharing is disabled. So
Well, that's a shame. It's probably something, a setting that I would have to make in Zoom.
yeah, I think when I've tried to do it during workshops, there were either 3 dots when I hovered over the share screen button, or I right-clicked on the share screen and it gave me an option to allow participants. And people are just gonna have to be on their honor that I'm the only one sharing.
Okay, there's the Zoom screen that I have, and I don't see any 3 dots.
No, it's on the bottom of, if he sets me up as a co-host, we could use the same whiteboard, but I'm not set up as a co-host. No, it would be in the main, at the bottom of the main window with everybody's faces, where there's a green button that says share screen.
Yeah. Got that.
Try right clicking on that maybe. I'm trying to remember what I did in.
Let's see. Get rid of that. Okay, when I right click, I get nothing.
Okay.
Okay,
Maybe.
have advanced sharing options. Maybe that's it. Oh, multiple participants can share simultaneously.
And then there should be something about enable participant screen sharing or something along those lines.
Advanced sharing options. Yeah, I've got that, like multiple participants can share simultaneously.
Okay, there we go.
All right.
Okay.
So now,
Everybody getting to see this now?
Yes.
Okay. So what we can start out with, this is going to actually be very similar to the question you already saw regarding test crosses. And this is the question. And this is a typical Bio 130 test question. So in guinea pigs, black fur is dominant over white fur. Now, unless otherwise specified, we're going to have to assume complete dominance, which means there's no blending, there's no expression of both traits. If one thing is dominant over the other, we only see the one thing. So in this case, the fur is either black or it's white because we're assuming complete dominance. How could an animal breeder test whether a black guinea pig is homozygous or heterozygous? So what does it mean if the guinea pig is homozygous?
He has two dominant genes.
Sure, he's got two dominant genes or two of the same alleles. And what about heterozygous?
He has one of each allele.
Absolutely correct. He has one of each. So we want to find out whether this black guinea pig is going to be homozygous or heterozygous. Now, the first thing you want to do is you need to define your variables. So what do we want to consider? I mean, we're looking at a trait for color. So what do we want to use as our variables here? I would suggest something simple like big B equals black, little b equals white. And does that match the capital letter here? Does that match what we would expect for a dominant trait? Is that the standard notation? Ingrid's shaking her head yes, and she is correct. And we've said that black is dominant, so this matches not only standard notation, it matches what you're given in the problem. White fur, if it's not dominant, it must be the recessive. Standard notation is we would use a small letter. Now, it's important that you use the same letter for each one, just one capital and one lowercase. If you're using different letters, now you're looking at something called a dihybrid. This is just a monohybrid that we're looking for. So remember, we're doing a test cross here. That's how you figure out whether something is homozygous or heterozygous. What is, so what does the genotype of the white-furred rabbit look like? What are the letters that we're going to use for the white rabbit?
Two lowercase b's.
Two lowercase b's, excellent. And how about for the black rabbit?
Either homozygous, like two capital B's, or heterozygous.
Yeah, so those are the letter combinations we would be looking for with the white rabbit or the black rabbit. So all we're going to do is lay out a quick Punnett square. And a Punnett square is simply a 4 box here, because a monohybrid only requires us 4 boxes. On one of the axes, all right, say this top one, we're going to represent one rabbit, and on this side, we're going to represent the other rabbit. So one of the things I would want to do would be to, it doesn't really matter which one you do. We can say we have a B and a B. All right, which rabbit is this?
The white.
The white. And then over here, we would actually end up setting up 2 different Punnett squares. It could either be that, or what else could it be? It could be a big B, or it could be a big B and a what?
A little b.
A little b. So we would really do 2 different Punnett squares if we were trying. The actual answer to this question is simply you would do a test cross, and that would be the end answer. But we're actually going to work this out just to make sure that you guys see why they do this kind of thing and why the information works out. So I'm just going to quickly set this up with a little b and a little b, and we said the one over here was going to be a big B and a what? This is the alternate, so a little b. Does everybody, honestly, does everybody understand why we set these 2 different squares up the way we did? Okay, silence is not helpful, but we're going to assume that everybody now understands that we're still doing a white rabbit crossing with the 2 different possibilities here for the black rabbit. So this is just simple, like almost math. What do I get if I have a big B in here and a little b? What kind of rabbit is that?
A black rabbit.
It's going to be a black rabbit. Excellent. What am I going to get in this box if I take big B over here and bring little b down?
Same.
Same. What am I going to get in this box over here?
The same.
Thing.
Okay. And over in here, we're going to get the same again, right?
Yeah.
Does everybody understand how we end up with this?
Yes.
Okay, so these would be all black rabbits. And what about over here? What am I going to get in this box?
Black.
A big B, little
Black.
b?
Yeah.
Okay. What am I going to get in this box?
Black
Same.
rabbit.
B, little b.
B, little b. What am I going to get in this box down here?
2 lowercase
White.
b's.
Yeah, I heard someone already say a color, going to get white. All right, and I'll get the same thing over here if I bring the big B and the big B. So if I have, if I wanted to know if this is homozygous or heterozygous, the black one, I would have to do this back cross. I would have to test it against something that was a known true breeding recessive. In this case, it would have white fur. And if I did this breeding and I bred 100 of these guinea pigs and I only ended up with black fur, I can be reasonably certain that I'm probably dealing with a homozygous dominant black furred rabbit. Because what's going to happen here if I breed 100 rabbits? What am I going to end up with?
Well, we don't know. Just mixed, right?
Well, we do know, because what is this big B, little b going to look like?
That one black.
And how about this one over here?
Black.
And how about this one down here?
White.
And how about this one down here?
White.
Okay, so I could have black or white. And it turns out that how many options do I have? I have 1, 2, 3, 4 different outcomes possible, right?
Yes.
Okay, so if I breed, another way of looking at this is 2 out of 4 options are going to be black and 2 out of 4 options are going to be white. Or what is that? Roughly 50%. Yeah, it's 50%. 2 of 4 and 2 of 4. Do you see where we're getting that percentage?
Yes.
Okay, so if I breed 100 rabbits, what should I probably expect out of this combination here?
50 white and 50 black.
50.
50
Yeah,
white
50,
and
50.
50 black, right? Is there any chance at all over here that I'm ever going to get a white rabbit?
Nope.
Nope. So this is why we do that test cross, because if the rabbit was heterozygous, there's a 50% chance that I'm going to have recessive traits come out in the offspring. But over here, if it's homozygous dominant, there's a 0% chance that I will have any white offspring show up. So this is why the test crosses work. And that's why they have to be done this way. Does that make sense to people? How we did this?
Yes.
Yes. This makes sense for me.
Okay, makes sense for you. So some keys to these problems are reading very carefully to see what they're telling you. First off, figure out what thing is dominant over what thing. Make sure that you identify in writing what your variables mean. Just like a math problem, if you don't identify your variables, the professor has no idea what you're talking about, and you can get yourself confused. So put them out here. Even when I do these nowadays, I still label my variables. I figure out what my genotypes are potentially based on the information given in the project. And then you have to just figure out exactly what the answer is going to be. They may ask you, what are the phenotypes that are possible from a homozygous? And so you have to know what's the difference between a phenotype and a genotype. We're out of time, but you need to know these little definitions, because if you don't read the question carefully, you might end up either answering it incorrectly or only answering it partially. So I can go over a lot more of this stuff during workshops for anybody that can attend. Happy to do it. There's lots and lots of examples and practices, and they're not very hard to do once you've kind of played with them a little bit.
Okay, thanks, Scott. There will be a quiz next Monday, and in all likelihood, there will be at least one problem like that on the quiz. But the quiz will be short, only 15 minutes, 10 questions as usual, and it'll cover what I went over today in terms of molecular genetics and what we went over last time and today in Mendelian genetics. So I won't ask you anything about molecular genetics that I haven't covered today.
Are you going to post the slides on Canvas?
Yeah, I'll put the slides on Canvas and I will send you, you've got the first worksheet. I'll send you the Mendelian worksheet with answers so you can check your thinking against mine. And I will send you another worksheet as soon as I make it up on molecular.
Awesome. Thank you, sir.
Okay. So you're going to have to rush to make it to Scott's workshop, which starts in 12 minutes. And if there's nothing else, I'm going to end this session.
I would ask for a moment of your time, sir, once everyone's gone.
Yeah, okay.